morning, dear colleagues. I'm thankful to organizers of this conference for inviting me to give a talk. The title of my today's talk is The Perineuronal Net and Synapse Geometry, Their Role in Brain Plasticity. Perineuronal net is a highly structured subtype of the extracellular matrix in the central nervous system that covers cell bodies and proximal dendrites of neurons in large neuronal subpopulations in the brain and spinal cord. It ensheaths synaptic terminals, like you can see in this picture, GABAergic synaptic terminals stained for vesicular GABA transporter we got in green, and perineuronal net stained with Wisteria floribunda agglutinin shown in red. This structure is especially interesting for its involvement in the regulation of brain plasticity. Why to study? Extracellular matrix provides spatial organization of molecular interactions and diffusion between cells on the cell surface between cells and on the cell surface. So all these effects, they do not happen like in solution voluntarily, but they are strictly regulated by the spatial framework provided by the extracellular matrix. During embryogenesis and early neuronatal development, massive synaptogenesis and refinement of neuronal network, synaptic network happens, and that is a critical period that is finished by the formation of the perineuronal nets on neuronal cell surface. So expression of perineuronal nets is induced by neuronal activity, by synaptic activity. In turn, formation of perineuronal nets finishes the critical period of massive synaptic network remodeling. This tight bidirectional connection between perineuronal nets and synaptic functions suggests that the structure of perineuronal nets must have a strong effect on synaptic regulation, but how it happens remains largely unknown. Over the last decades, there has been a massive progress in molecular biology studies on perineuronal nets, in electrophysiology studies, demonstrating behavioral importance of the perineuronal nets in the brain function, but the microstructure remains the quantitative studies of the perineuronal net microstructure remained essentially absent. And as this field is quickly developing, when we started these studies, maybe the only pioneering work on the quantitative study of the microstructure of perineuronal nets was by the group of Alexander Detyatev, published in 2014. And then recently, a couple very interesting papers came on microstructure, super resolution studies of the microstructure in the model of focal cerebral ischemia, and then in the model of sensory input deprivation for the visual cortex, and a model of Rett syndrome. A brief overview of PNN functions and related pathologies. Most importantly, most studied, PNN is involved in the regulation of neuronal plasticity, restricting remodeling of synaptic networks, synaptic plasticity. For instance, PNN restricts motility of AMPA receptors on neuronal cell surface, cation buffering. Perineuronal nets are strongly negatively charged because of chondroitin sulfate proteoglycans, a major component of perineuronal nets, making it a buffer for cations. And also cell surface distribution of signaling proteins. Over recent years, PNN was implicated in pathogenesis of epilepsy, oxidative stress, Alzheimer's disease, post-traumatic regeneration, schizophrenia, and stroke. These were our first results on quantitative studies of the microstructure of perineuronal nets. This is, we first did it on the mouse and rat somatosensory cortex. So because quantitative description was essentially totally missing, we started with measuring simple geometry of the meshes of the net, and we demonstrate that, for instance, pentagons is the most frequent geometry shape of the meshes, and we also demonstrate that the size of each mesh can vary very widely, and even in neighboring regions of one neuron cell surface, the area of individual mesh can vary 10 times. First we did it in 2D single confocal planes, and then we were asked by reviewers to do it also in 3D. We performed three-dimensional reconstructions of confocal stacks with filament tool of MRIs, and we demonstrated that our measurements performed in two-dimensional analysis are essentially confirmed by the three-dimensional analysis. Intuitively, we can expect that the size of a single synaptic terminal restricted by the surrounding perineuronal net would influence the strength of synaptic input. That remains to be proven in case of the synapses surrounded by perineuronal nets. And this raises a few interesting questions. What functional consequences are determined by the size of the synapse mesh complex? Do meshes change their size adapting to the synaptic network state, or do they keep their size constant? Do the smallest meshes represent functional synapses? Are they newborn meshes that are going to extend to a larger size, or did they shrink from a larger size for a functional adjustment, or because the corresponding synapses were eliminated? Then we looked at the distribution of the chondroitin sulfate staining intensity signal along the single mesh perimeter. And we observed that there are two distinct types of the mesh microstructure. There are meshes with chondroitin sulfates enriched in the vertices of the mesh. Here you can see a plot of the intensity, VFA staining intensity distribution along the mesh perimeter, and you can see that maxima are in vertices. And there is also a different type of meshes where the intensity signal is more or less randomly distributed along the mesh perimeter. So we called this kind of meshes as polar meshes and these as non-polar meshes. We suggested polarity index as a quantitative measure, essentially a ratio of the intensity in vertices to intensity in middle parts of the edges. Like here are intensities in vertices and here in the middle parts of the edges. We show that there is similar distribution of the polarity index for mice and rats. Again, we started with two-dimensional analysis and then expanded to three-dimensional analysis. And 2D and 3D measurements correlate. Here you can see a 3D tracing of a single mesh with vertices marked as green balls and middle pixels of edges marked as magenta. And here is distribution of VFA intensity, surface reconstruction with Imaris for the three-dimensional distribution of the staining intensity for a single mesh. Then we looked at the distribution of polar and non-polar meshes on the cell surface, and surprisingly we found that polar meshes shown here in yellow dots, they cluster together, and non-polar meshes also cluster together, so they are not randomly distributed. That is observed in both in 2D and in 3D analysis. Functional significance of this finding remains to be investigated. We can guess that this may be an additional mechanism of functional compartmentation of the neuronal cell surface. Then, from studying just perineuronal nets, we go further to the studies of the complex of perineuronal net with synaptic terminals. So you can see terminals of GABAergic synapses stained for VGAT here as green punctum. So we can get quantitative values for the geometry of this complex. Here is an example of a mesh with a synaptic terminal inside. We make image segmentation via auto-thresholding with Fiji software, and besides we can make 3D reconstruction of this PNN synapse complex with Imaris. So in C, D, and E, you can see side projection of the complex with synaptic terminal surrounded by the perineuronal net. And in F and G, you can see XY projection of the complex. We applied this approach to study the effect of sensory input deprivation on the structure of the synapse plus PNN complex. So we performed somatosensory deprivation by whisker shaving in newborn mice. And we demonstrate that by the age of 1 month, the synapse shape is affected. So under synaptic deprivation, the area of VGAT positive puncta becomes significantly reduced as compared to control. To further improve the resolution of the microstructure, we use SCON super resolution microscopy that allows improving spatial resolution about twice in XY direction. So you can see two types of the complex morphology with complex synaptic puncta. We got positive synaptic puncta surrounded by PNN, and more flat and larger area synaptic puncta. And in addition to single mesh and very high resolution studies of the single synapse structure, we also needed a method to make wide-span quantitative characterization of perineuronal nets. That would work for thousands of neurons in large tiles, microscopy tiles of brain sections. So we developed a method to study single cell PNN area and intensity for thousands of cells in large tissue section microscopy tiles, taken with 10 times, 20 times objectives. Here you can see these two neurons magnified here. This is a tile of a somatosensory cortex section of an adult mouse. And this is the result of segmentation, where you can see the perineuronal net segmented, allowing for the area and intensity quantification. For that purpose, we essentially choose a single cell and apply several sizes of the analyzed region. For each size of the analyzed region, we perform auto-thresholding segmentation with 16 different auto-thresholding methods. And then we can choose, for each particular neuron, we can choose the optimal area. In this particular case, it would be either I or J. And then recently another method appeared for the same purpose, allowing to quantify the PNN intensity in large numbers of neurons in tissue sections, and it has been demonstrated for the model of cocaine treatment and its effect on medial prefrontal cortex. And what we have done, we studied both PNN development during first postnatal month of the mouse in somatosensory cortex, and in this picture you can see our studies on the spinal cord, on the cervical spinal cord, where you can study again thousands of single cell perineuronal nets, segmentation shown here. You can segment the tissue to quantify the parameters at different spatial points along the spinal cord. This is distribution of PNN area and intensity along the cervical spinal cord. And we applied this approach to look at post-traumatic changes to perineuronal nets in the model of the cervical spinal cord hemisection. That makes, for instance, important effect on the respiration control and poorly regenerates after injury. Here you can see the injury site surrounded by the spinal cord tissue with perineuronal nets. You can see the redistribution of the PNN area and intensity around the injury site, along the cervical spinal cord. To summarize, current data on the cell surface signaling suggest that the perineuronal net spatial structure is likely to have a profound impact on synaptic function. But this impact and its mechanism remain essentially unstudied. We are aiming at revealing the PNN structure-function relation by combining low-resolution tissue imaging with single synapse super-resolution structural studies and quantitative image analysis of the PNN microstructure. So I would like to thank my collaborators at Kazan Federal University, at the University of Helsinki, and at my current position in Denmark. Thank you very much for your attention.
Thank you for a nice presentation. Any questions?
So when you analyze the intensity and you separate different subset of the cell, polarized and unpolarized, what is the criteria for you determine the polarized versus non-polarized?
Maybe I go back.
Yeah.
So look, this mesh with intensity distribution shown in detail here and here. So the intensity is enriched in the vertices. And if you just quantify simple ratio of the fluorescence intensity in vertices versus middle points of the edges, you get what we call polarity index that will be higher than 1 for polarized meshes and will be around 1 for non-polarized meshes.
So all based in Murray's analysis.
No, this is Fiji. This is Fiji analysis, but this is a Maris analysis that gives essentially the same.
My question is, so as I understand, you are just measuring the fluorescence of two different types of compounds, which bind two types of fluorescent dyes.
That's what I present.
And the question is, what are there any data regarding chemical nature of the compounds they are studying?
So the a red signal here is for chondroitin sulfates. Those are sugars, highly negatively charged, and there have been extensive studies of their chemistry and importance of chemical composition for physiological functions. And for the green signal, that is vesicular GABA transporter, so that is a protein that pumps GABA from cytoplasm into vesicles, and it's antibody staining. So it essentially doesn't tell you about chemical structure, it's an antibody antigen binding.
Thank you very much.
Thank you.